this is appropriate that I'm bringing Dr. Artigas over here to talk. And we haven't quite got to the functional aspect of it because we want to get to that today in today's lecture and talk about membrane transport. But first of all, we have to finish talking about just the basics that you've already been exposed to. And this thing, of course, is called the fluid mosaic model of lipid bilayer. Why is it called a mosaic? What creates a mosaic? And so where are the mosaic pieces here? Well, the proteins. Okay, so originally the intention of that model was you have all these little pieces that fit together to create functional groups or whatever those things do, and that was the mosaic part. But we now know it's not just the proteins that are a part of the mosaic, it's the lipid itself. So this looks like a homogeneous group of lipids. That's actually not the case. You can have, if this is fluid, that means we've actually, the temperature is above the melting point, so this becomes very fluid, right? And so actually these phospholipids, which they can fuse in 2 dimensions, this way and that way, but they can also spin around, so they vibrate at high rates of speed. In fact, it only takes one of these guys about 20 seconds to go around the entire surface of the cell. So it's very fast, so it's very fluid. But everything is not like that. There are regions that are actually in a second order, second state, and that is the lipid rafts. So you have the SOL, S-O-L, means the soluble fluid state. Okay. On the other half of that, you have the gel state. So there's a transition between gel and sol. So those are the 2 basic components. But you've got an intermediate thing, and that's called liquid ordered. So that's the state in between gel and sol, liquid ordered. And so you have patches, or like you can think of them as little icebergs made out of lipids that are thicker and less fluid than the rest of the membrane. What creates a lipid raft? Well, what creates a lipid raft is it collects certain components that are in higher concentrations. So what's higher in this lipid raft is, number 1, cholesterol. Cholesterol. Anytime cholesterol is concentrated, it makes the membrane thicker. So that's one of the functions of cholesterol, is to thicken the membrane. It also recruits a particular class of phospholipids, or not phospholipids, but membrane lipids called sphingolipids. And you need to go back and glance at what a sphingolipid, how it differs from a traditional phosphoglyceride. It actually does not have that 3-carbon glycerol backbone. Instead, it has a sphingolipid backbone. And the fatty acid tail on a sphingolipid is very long and saturated. That means it's very straight, okay? So the second reason why this is thick is that the lipids are long and straight, and they actually interdigitate with the other leaflets. So this is the exoplasmic leaflet, this is the cytoplasmic leaflet. So it actually interdigitates so that what happens is both leaflets are coupled together. So the first coupling method that makes those 2 leaflets sort of stick together and move as a single iceberg is the length of those sphingolipids. The second point is it recruits particular proteins, particularly signaling proteins. And those transmembrane proteins help couple the 2 pieces of the iceberg together. Okay, so there's protein coupling and lipid coupling are what makes this thing stick together. And it's like a little iceberg. It floats around, and so you can actually see them using certain types of fluorescent microscopy. Okay, so this is a sort of a second state of matter between sol and gel, liquid order. And the whole point of this is to recruit different proteins together as a functional group. And it has things to do with signaling, okay, particularly signaling molecules. But the other type of protein that likes to sit in this little lipid raft is lipid anchored proteins, and we'll talk about that class of membrane proteins today. Okay, so 2 things contributing to the mosaic: lots of weird proteins, flying around, and also these little special regions of rafts moving about. So it is not homogeneous. Okay, so let's go into the categorization, or quickly look at the different categories of membrane proteins. Deal with the membranes first, and then we'll talk more about the lipids. Okay. So here's the different classes of membrane proteins. We'll start with this one here, which is a single-pass membrane protein, and the poster child for that type of protein is glycophorin. It's one of the first membrane proteins that was isolated, and so the motif that it uses to get across the membrane is a single, or actually it's a secondary structure, not really a motif, is a single transmembrane alpha helix. Okay, and this needs to be long enough to get across that lipid bilayer, which that means it takes about 20 to 25 amino acids, sometimes a little less, sometimes a little more, in order to span the distance across that lipid hydrophobic area. And so what would you predict would be the nature of those R groups coming off radially that are buried into this lipid? It should be hydrophobic, right? That makes lots of sense. In fact, we have a technique called a hydropathy plot that we'll look at, and you can actually look for these using programs. Just by looking at the sequence of a protein, you can ask, well, is this a hydrophobic membrane spanning alpha helix or not? There's a way to do that. So we'll look at that technique later. So this is the extracellular domain, and there's two reasons why you know that belongs in the extracellular domain. Number one is of all the disulfide bridges. Disulfide bridges are not oxidized unless they're facing either outside the cell or the luminal compartment of the secretory pathway, which is ER, Golgi, vesicle, membrane. Okay. So exoplasmic compartments favor the formation of disulfide bridges, and you use this to stiffen the protein because it's now exposed to the outside of the cell, which is, it has to protect itself against all kinds of non-specific proteases, changes in salts that are not being controlled like they are in the cytoplasm. So it gives it some structural integrity. And then you see the sugars. Sugars are almost exclusively on the exoplasmic surface. Okay? And the sugar coating, again, helps to protect the protein. It also is involved in signaling, so there's lots of different functions, but almost exclusively, you can tell if this is the extracellular domain of the protein, that's where the sugars will be attached, and that's where the disulfide bridges will be attached. Now, is it possible to have cysteines reduced in the sequence? Absolutely. I'll show you one particular function that you can use this sulfhydryl group for to attach lipid anchors to. Okay, so here we go. We have this single-pass membrane protein, and so that's a good example of that group. Now, all you have to do to get it to the next class is to put multiple alpha helices, and this is very typical. This is the very first multipass membrane protein that was isolated. It is a bacterial protein. The only reason I use it is your book talks about it a lot. Okay, so it's bacterial rhodopsin. It's got 7 transmembrane alpha helices. And you notice they usually show them as sort of laid out in 2 dimensions, but they don't assemble that way in the membrane. They actually are sort of gathered together like a 6-pack of Coke or something. Certainly not beer, but it's a 7-pack actually. And it creates actually a chamber. And what happens is a photon of light strikes this retinal group, causes a shift in the protein so that a proton can just snake through this little hole. Okay. So this is a proton pump. It's actually pumping out protons, creating a proton energy gradient that the cell can use to suck things back into the cell using proton symport with a, say, amino acid or a sugar molecule. So it's a way of producing an energy gradient directly from light. So it's kind of cool. Where do we encounter those in our proteins? Membrane transporters are almost exclusively multipass membrane proteins. They're almost all that way. Okay. So that's the second class. Then we have the third way to get across a membrane is to use a BetaBarrel. So it's rare in eukaryotes to use a BetaBarrel, but the one place that's really not eukaryotic in us is what? The mitochondria and also chloroplasm. And so this is the outer mitochondrial membrane. So this would be This would be the cytoplasm, this would be the intermembrane space, and then there would be a second membrane down here where all the electron transport chain is located. But on the outer membrane, you want those nutrients to be able to diffuse easily up again into this intermembrane space, and so you create this circular beta barrel motif that creates a hydrophilic pore, so things like pyruvate can just go right through, and then they're transported across the inner membrane and turned into ATP. Okay. So this is there's really only two ways to get across the membrane. Alpha helix, that's about 20 to 25 amino acids, or this motif here, which is a beta barrel. Okay. That's known as a porin. Okay. Next kind is an embedded protein. There are actually, we now know several examples of this type of protein, but until a few years ago, this was the main example. Okay, so it's an alpha helix that only inserts into one leaflet. That's its hallmark. Okay, and it doesn't insert vertically. It sort of kneels into the leaflet like this. Okay, so it's an alpha helix that's oriented parallel to the surface of the membrane, but it's actually embedded in the hydrophobic layer. Okay, and some of the R groups that point out those are going to be hydrophilic. The ones that face the hydrophobic interior are hydrophobic. Okay, and so a good example of a protein that a poster child that would represent this group is prostaglandin H2 synthase. Okay? And prostaglandin H2 synthase is really important because it controls the production of prostaglandins. And prostaglandins have good, bad, and ugly functions. Okay? The good functions for prostaglandins are that they keep platelets, they have a role in platelet aggregation. Okay. That's one of the reasons you take aspirin, is because you inhibit the ability of the prostaglandin to aggregate the platelets, and so you have better fluid flow through your cardiovascular system. So, but when you need clotting, you need prostaglandins, certain types of prostaglandins. So it's involved in clotting. Prostaglandins are secreted to protect the lining of your stomach. Okay, so prostaglandins stimulate mucus production that keeps that really low pH from chewing into your gastric mucosa. Okay, that's why aspirin is bad, because aspirin is a non-specific inhibitor of all prostaglandin synthase molecules, and so it turns off the secretion of that mucus layer and exposes your stomach to that acid. That's why you want to take enteric-coated aspirin, so it goes all the way into your small intestine before it's dissolved. That's what that coating will do for you. Okay. Other good things I'll just mention in passing. Of course, I go into a lot of detail in my endocrinology course. It's responsible for controlling ovulation during the menstrual cycle. It's also regulates parturition, that is birth. Okay, so lots of really important functions. Okay. Now, are there bad functions? Yeah, the reason you take aspirin typically is to get rid of a headache. And so there's really two types of enzymes that are involved in making prostaglandin. So let's say, here's the good one. Sorry, let me get this on. Okay, so let's say the good, and then here's the bad. So all those good ones that I just got through talking about are actually made by a particular enzyme called cyclooxygenase, and we'll just call it a COX enzyme. And so COX-1 is the good one. And it has a particular shape to it. This protein is shaped with a fairly narrow entryway. And so this is where the substrate fits into that. Right? And then the bad ones are mediated by the COX-2 enzyme. And it has a fairly similar shape, except it's got a much clunkier looking opening. Okay? Now, the problem with aspirin is aspirin will fit into here, because aspirin's like a little sliver. And so the aspirin will fit in there, but it also will fit in here because it's a big gaping area. So aspirin inhibits the good and the bad together. Now what somebody did was come up with a way to make an inhibitor called Celebrex, which is a specific COX-2 inhibitor. So you want to inhibit the bad cyclooxygenase, and that shuts down prostaglandin synthesis. And you can see this big fat boy will fit in here, but it won't fit in the COX-1 binding site. And so that's a really good example of a drug that's a competitive inhibitor. And you want it to be competitive, right, and not poison the enzyme, because that enzyme is there for a purpose. And so you just want to knock it down until your headache goes away, and then you get rid of it. Right? Ibuprofen works the same way. Except, well, ibuprofen is another general cyclooxygenase inhibitor. This new class of drugs, there are actually some problems with it. The side effects were that it inhibits certain aspects of heart valve function. So they've had to rework on this problem, and this is a really nice way to attack arthritis and various inflammatory diseases. You can specifically hit the COX-2 enzyme and leave the COX-1 alone. Okay? Whole point of this is the enzyme that actually, not the COX enzyme, but the prostaglandin H2 synthase, which is in that same pathway, is used to make prostaglandin, and it's a embedded protein. Okay. Questions about that kind of protein? All right. That brings us to the lipid anchor proteins. Okay. And I forgot to mention the two big broad categories, and we're talking about one category, and that's the integral membrane proteins, is they're differentiated from peripheral membrane proteins in the following way. If you can treat the membrane with high pH, high salt, if you remove the calcium, that is, you put a calcium chelator in there, anything that will disrupt the exterior proteins, those proteins can be pulled off. Those are called peripheral proteins. But if you do those nasty treatments to an integral membrane protein, single pass, multipass, porin, lipid anchor, none of those can be pulled out of the membrane without the use of a detergent, because they're integrated into the surface or the width of the plasma membrane. So if it comes out with just a salt treatment, it's a peripheral membrane protein. But if you have to treat it with a detergent, and that's what's required to pull it out of the membrane, then it's an integral membrane protein. Okay, so this is the fourth class of integral membrane proteins, and that is lipid anchor. So here's the protein up here, and let's talk about RAS first. I think that's what we use in the notes up here. Yeah, RAS is a signaling molecule that you'll get real familiar with. It tends to be overexpressed in lots of different kinds of cancer because it is a proliferative pathway. If you activate the RAS pathway, you turn on cell division. Okay, and we'll talk about that process. But RAS is actually one of those small GTPase molecular switches that I was mentioning last time, like the eukaryotic initiation factor number 2. This is in the same family. But the way it sticks to the membrane is it has a lipid anchor, okay? And not one lipid anchor, it actually has two. Each one of these lipid anchors is fairly weak, okay? So here's the lipid anchor. This is a cysteine, and this representation of the membrane is reversed from what you were just being shown. The cytosolic compartment is up on top, the exterior is down underneath, okay? So these signaling molecules protrude into the cytoplasm, and that's how they convey the signal to the inside of the cell. But the way they're hanging onto the membrane is this sulfhydryl group is actually attached to a rubber tail. It's actually made out of isoprene units, which some of you may remember from organic chemistry is the unit that you make rubber out of. It is each isoprene unit is 5 carbons, and if you stick 3 of those together, you get a rubber tail that's about 15 carbons long, and that's long enough to make his tail insert into 1, not 2 leaflets. So it's not a membrane spanning rubber tail, it goes into 1 leaflet, okay? And that is the cytoplasmic leaflet. Okay, so these isoprenyl, or sometimes we shorten it, we just call them prenyl tails, is one way to hold RAS into the membrane. The second way is by a 16 amino acid fatty acid tail, which is known as palmitic acid. And so palmitic acid uses the same type of thioester bond to that sulfhydryl group. So now you have a new function for a reduced sulfhydryl group. You can attach lipid tails to them. So it is the case that you have exposed cysteines playing a role in the structural integrity of membrane proteins, even though they're on the inside of the cell. All right, so it takes two of these to make a RAS hold on. And then the second example is an old friend of ours. SARC is a lipid-anchored tyrosine kinase. And again, the same rule applies. One of these tails is too weak, okay? It uses a myristate tail, which is, the only difference between palmitic acid and myristic acid is this has got 16 carbons in this fatty acid tail, this one has 14, okay? So it's holding on this way, but your book doesn't show you, but it's also got a second palmitic acid group. So it shares in common this one with RAS. But the two of these proteins are really overexpressed in many types of cancer. So there's lots of drugs that target these proteins, anti-tumor agents. Okay, any questions about that? So this is two types of, so let's make sure we understand the category. This is a type of integral membrane protein, even though the protein is actually not touching the membrane. It's holding on with this lipid tail. The third type is also biologically important. Okay. And that is this ugly term. These are GPI-anchored proteins, and that stands for glycosyl phosphatidylinositol anchor. And that's why we call it GPI, so we don't want to strain ourselves. But it's got a phospholipid that's inserted into the membrane. Here, let's get a good picture of it. Let's see. Where did it go? Go back. There we go. This is it, sort of a beast-looking protein. So it's got two phospholipids, just like a standard phosphatidyl anything, okay, two lipid tails, but they're part of a phosphoglyceride backbone. Okay, so this is not like a single tail. It's got two of them. They're always inserted into the exoplasmic leaflet. So this thing is going to be on the outside of the cell. So it's like a little kid standing with two legs, two fatty acids in the leaflet, okay, holding a balloon. Okay, the balloon is the protein. Okay. What's the link? His arm is actually this is sugar chain, and then phosphoethanolamine, and then this connects to the protein. So you don't need to know the explicit structure of this connection here, but understand that the protein is outside the cell in the hydrophilic environment, waving around. And these things, there's several different examples. One of them is an intestinal enzyme called alkaline phosphatase, poster child, alkaline phosphatase, okay? Alkaline phosphatase operates in your small intestine to cut off phosphate groups. So you eat a lot of stuff that's got phosphates attached to it, like what? DNA, phosphorylated proteins, lots of sugars that are phosphorylated, okay? Your gut does not want to transport a negatively charged thing across your intestinal epithelium. So it chops off the phosphate and uses this very specific phosphate transporter to pull it in, because phosphate's necessary, right? You use phosphate to do lots of stuff. So you want the phosphate, but you don't take them in at the same time. That leaves the protein or the lipid separate so it can be transported. So that's the plan of the intestine, is to break down everything into small monomeric pieces and then pull it across. Okay? And one of the things that does that is alkaline phosphatase. And so this is the enzyme, and it's waving around in your gut lumen, hacking the heck out of all those phosphate groups, removing them. Okay? So a second type that's biologically important is the prion. A prion is the infective agent protein in mad cow disease, and then the human version is Creutzfeldt-Jakob disease. And so prions are really nasty, but prions are actually, they use this linkage to get into the surface of neurons and screw things up. Okay, we have normal prions, and then there's the mutated type that is the infective agent in mad cow. All right, so prions are in this group. What's the last one? Let's see. Oh, this one here. Okay, so there's variable surface glycoprotein. Now, this is a protein that a protozoan, Trypanosome brucei, uses to actually fool our immune system. So what it's got is this little balloon waving around on the outside of the surface, okay, and it's heavily covered with sugars, and so the immune system is just all over it. It's trying to attack that parasite. And this parasite causes African sleeping sickness. Okay? So why doesn't the immune system just clean it up? Well, what happens is when the immune system starts to attack this protein, the trypanosome just releases the balloon and expresses a different protein. So it's a cloaking device, right? Now the immune system can't see it because it's attacking that other coating that's no longer there. So it's the Romulan airships were not the first cloaking device. The trypanosomes were. All right, all three of those examples are poster children with GPI anchored. The last thing I want to tell you about GPI anchors, they tend to associate into lipid rafts. Okay, they tend to pull together. All right, any questions so far? I think we've almost finished we have finished all the integral membrane protein types. Now, we go to the peripheral. These are a little bit easier to understand. So the peripheral, there's two types of peripheral membrane proteins. They're the kinds that just, actually it's not even shown in this picture. This is an example of an indirect peripheral membrane protein. And that's because it's touching the membrane because it's bound to an integral membrane protein. So it uses some other protein as a Klingon to grab onto. That's an indirect peripheral membrane protein. And there's lots of examples, and I'll show you those here in a moment. The other one is not shown on that particular slide. Here we go. Okay, so the indirect method this way, because it's holding on to this single pass glycophorin-like thing. The other way is actually the protein directly binds to that hydrophilic surface. Remember I said that the surface of the membrane is bumpy, and there are certain proteins can actually recognize the surface and bind to things like, these are like phosphatidylanosyl inositol residues. This protein called annexin will actually bind to those phosphatidyl serines, the head groups, right? And so this is a direct peripheral membrane protein. But if you expose this cell to high salt, high pH, urea, nasty things, this thing will come apart. The non-covalent bonds will be broken, and then you will solubilize the peripheral membrane proteins, hence the term peripheral membrane protein. Okay. What do you need to pull these guys out? Detergent, right? Let's see if I have a particular. Oh, okay. So that annexin was an example of a direct peripheral protein. This is an example of an indirect. So this is a protein called band3, and this is the inside, the cytoplasmic side of what a red blood cell looks like. Okay, it's really complicated. And let's just zoom in on this particular structure. Band3, they call it that because they lacked imagination back then. This protein is a chloride bicarbonate exchanger. Okay, so it's an antipore. It brings in bicarbonate and it gets rid of chloride, and it can actually work in the opposite direction. So this is a secondary active membrane transport protein, and as you now know, what should it look like? What type of membrane protein will it be if it's a transport protein? Yeah. Well, this one, since it's secondary, so that's kind of a tricky question. It's running off a gradient from that somebody else that a pump created. So this is secondary active. So it is an active transport. But let's just say if it's a membrane transport protein, what's it going to look like? Multipass. So that's what I want you to think immediately, that this two barrels are going to be multipass membrane proteins. Okay, so that's an integral membrane protein, but you can stick a peripheral membrane protein on it, and that's what you do with anchoring. And anchoring tells you exactly what it's doing. It's anchoring this cytoskeletal array on the inside surface of the red blood cell. That strengthens the red blood cell and enables it to go through single file convection through your capillaries. That's a tough squeeze. And so it puts pressure on the red blood cell, and if it didn't have this supportive network of proteins, it would burst, and you'd be anemic. That's what would happen, okay? So this is strengthening the inside layer of the red blood cell so you can deform it without crushing it. But that is a really good example of a indirect peripheral membrane protein. And everything attached to it also qualifies, right? So this looking thing, you actually saw that in the video on the first day. It was kind of, you saw these little spaghetti looking arrays. That protein is spectrin. And spectrin binds to the actin cytoskeleton and strengthens the membrane system. But all of these proteins are binding to the membrane because they are indirect peripheral membrane proteins. You add salt to this, and all of those are going to fall apart, except for that guy, and that one would stay in the membrane. Okay. Yeah. Yeah. No, band 3 is the multipass. The peripheral, well, it's that yellow guy and everything else that's stuck to the membrane. Because this is the only guy that's actually. I'm sorry. The membrane is this sheet. Those are lipids, right? Okay. And then these are the mosaic proteins that are moving around. Okay. The other thing, I've already mentioned the fact that sugar groups are sort of diagnostic. They're going to tell you which side of the transmembrane protein is facing outside and which side is not. So if the sugar's on this domain, that means that domain faces out. And if there are disulfide bridges on that particular domain, let's say you isolated this protein, how could you tell which side of that transmembrane alpha helix is facing out and which side is facing in? By the residues that are attached to it. So if there's sugar stuck to it, they're going to be facing this way. If there are no sugars and no disulfide bridges, it will tend to be this side of the protein facing in. So you can actually tell which way the transmembrane alpha helix is pointing. Okay. I think let's take a break there, and then we'll go into some techniques. Dynamics. A couple of techniques are real standard. One of these is freeze fracture electron microscopy. I don't think it finds as much use today as it used to, but it is a good way of looking at the aggregation of proteins. So for example, if you isolate this patch of this bacterial, it's called a purple membrane, there are so many bacterial rhodopsin molecules that they aggregate into this array. And you can actually see that array if you do freeze fracture. And what freeze fracture does is you basically freeze the cells at very cold temperature, and I'm talking like minus 180 or even lower, and then you crack it with a diamond knife, and it actually sends a crack through the cell. And the fracture lines tend to go through in between the leaflets of the membrane. So you peel them open, and you can actually see a little model of where the proteins were inserted into that membrane. And so that's what you actually have to do a lot of stuff. You have to cast it with metal coating and then evaporate it and do all this jiggery pokery. So it takes a lot of manipulation, but you can't look at the distribution of proteins. And that's about what is it aggregated here or non-aggregated? And so that's the type of information you get from freeze fraction. Okay. A much more standard technique is SDS, PAGE, polyacrylamide electrophoresis that you'll actually do in cell lab. And most of you probably have this in some other class, but here's the basic principle. You have this gel, which is made out of polyacrylamide. And depending on the concentration of polyacrylamide, you'll actually be able to resolve different sizes of proteins. So the rule is, the lower the concentration of polyacrylamide, the bigger the protein is you can resolve. Okay, so if you have a 5% gel, you can only resolve things that are really large, right? So I think anywhere from, let me see what my data says. So like a 5% gel will resolve between 60 and 250 kilodalton in protein. So you use a 5% if you want to separate really large guys from each other. But what if you have a bunch of small, say, peripheral proteins? How do you resolve those? You increase the concentration of the gel. So at 15%, what's happening is the sieve, or the pore size, is really tiny, and that can resolve a protein as small as 10 kilodalton from a protein that's 50 kilodaltons. So higher the concentration, the greater the resolving power between small proteins. Okay. Now, how is a polyacrylamide gel resolving proteins to begin with? So actually, you have this gel, and that's got the pores that the protein is going to move through. What makes them move? Well, you put an electric field across this gel. So you have an anode down here, a cathode up here. And so the anions, the negatively charged things, are going to move towards this positive pole called the anode. So anions go to the anode. And what you do is you make sure that everything gets a negative charge. Are all proteins negatively charged? No. Okay. Well, we're going to make them negatively charged because that's where the SDS detergent comes in. This is the sodium dodecyl sulfate. And the reason it's a good detergent is it's hydrophobic at one end and it's amphipathic. That is, it's got two different natures. It's hydrophobic here and ionically charged there. It's negative. Okay. So what this thing is going to do is have two functions. It's going to pull out the protein from the lipid, okay, and then it's going to uniformly coat the protein. That will give you two really important properties. Everything is going to be rod-shaped because it completely denatures the protein. Okay, so now you've changed the shape of all the proteins. Some could have been alpha helical, others could have been globular. You change them all to rods. You change them all to rods so they have a uniform shape and they have a uniform charge density. That is, the charge to mass ratio is the same for every protein. Because the bigger it gets, the more SDS sticks to it. So you're adding, as it gets bigger, you add more SDS molecules. And that's what it looks like. So the SDS is going to pull it out of the membrane and coat the protein, stand it up into a rod. And so what you have is everything is rod-like and everything has the same charge to mass ratio. Now, why is that important? The reason that's important to give everything the same charge density is that proteins migrate through those pores based on three properties: the shape, the charge density, and the molecular weight. You've just taken out two of those properties. So if everybody has the same charge density, everybody has the same shape, what's going to be the driver of moving through the pores? Molecular weight. So everything will move based on its molecular weight. And what is resolving two different proteins, a big one and a small one, why is the small one going to move faster through the gel? Everything else is equal. We've got the same pulling power on both those two proteins. One's a little bit bigger than the other.
Because if you put a force on something small, that'll make it accelerate faster than on something large.
But they got a uniform charge density. So what you said would have worked. The smaller one would encounter a different pulling power, unless the charge density is the same on both pieces. So that can't be it.
Diffusion.
What's doing the resolving? The gel, okay, is the pore size. You've got a standard pore that you've chosen based on the percentage of polyacrylamide that you put in there. You've got two proteins trying to go through the same pore. Who's going to win? The smaller. It's going to go through faster because it's smaller. The pore is setting the limitation on how fast the protein can wiggle through there. Okay, so it's all based on molecular weight, but what creates the resolving power is the gel percentage, because that controls the pore size. Okay, and so what you get are a bunch of bands like this, and so that really non-creative person that named band 3, it was the third band that he saw on the SDS page gel. Okay. And there's two types of gels. What other problem do you have if you want to stand a protein up into a single rod? Remember, membrane proteins have other groups attached to them. Okay. Let's look at this guy. In fact, this was one of the first ones, and they had all kinds of problems getting it to run properly on a gel. Why would that be? I mean, you can pull it out of the membrane, right? You can stand it up, but what's wrong with this? It's got all kinds of disulfide bridges. You have to reduce those. And so you have to use a reducing agent, beta-mercaptoethanol, to break all those things, and then it will stand up straight. Except it won't, because of this. That's going to add a lot of mass to it, so you need to strip off all the sugar groups by exposing it to glycosidases. Those are enzymes that chop off sugar groups. And once you've done that, and if it had a lipid tail, you'd have to whack that off too. And there are lipases that will do that, or that will attack that phytoester. So you have to kind of know a little bit about your protein, or at least put all of those. If you didn't know anything about it, you could at least put all those enzymes and those conditions and treat it as a beginning to get to a point where you're pretty sure it's traveling as a fine upstanding rod instead of this mess here. So that's the difference between a reducing gel and a non-reducing gel. Is a reducing gel has the beta-mercaptoethanol, and you've denatured the protein. But if you want to look at how it migrates with those disulfide bridges intact, you don't use a reducing gel. Yeah.
Does getting rid of all those, all that stuff, affect the weight?
It affects the in situ weight, but if you're just looking at the protein, you want to know how fast the protein, or how big it is. Oh yeah, these things all add, well, except the disulfide bridges don't. But the sugar groups, this huge increase in molecular weight. Okay. Good. Okay, so that's a real, you'll do that if you take the cell lab, and let's do that. Okay, so let's say you want to know, so you're a bioinformatics person and you've never set foot into a cell lab. You can tell by looking at the sequence of a protein, you can query that database and ask, are there any things in that sequence that look like alpha helices, transmembrane alpha helices? And you've got something on your side, you know those alpha helices should be a certain length, 20 to 25, that's the general rule. And the nature of the R groups projecting radially off of those, that alpha helix should be hydrophobic because it's buried in the membrane. And there's some problems with that idea. But let's say we're looking at glycophorin, okay? And this is so what we're going to do here is measure something called the hydropathy index. And each, call this one. So each amino acid is associated with a particular hydropathy measurement, okay? The more hydrophobic it is, the more positive it is. Okay? And the reason those numbers are positive for hydrophobic amino acids and they're negative for all the polar and charged amino acids is the following. So this hydropathy number is actually the free energy it takes to move, when you move that amino acid from an oil, like olive oil, into water. So you're not changing the amino acid, you're just moving it from one solvent to the next. And there's a free energy change involved in that. And if you're hydrophobic, is that going to be something you want to do, energetically favorable? If you're in olive oil, you want to stay in olive oil, right? But if they move you into a hydrophilic environment like water, what's the free energy change going to be? It's going to be not very favorable. It's going to be positive. Positive free energy changes are energetically expensive. But if it wants to go there, like if good old histidine is sitting in olive oil, it would rather jump into the water. And so it has a negative favorable free energy change. So these all have this hydropathy index for each one of these amino acids. Okay, so how do you actually do the experiment? Well, you have to know the sequence, and you have that back here. Okay, so this is the sequence starting at the N-terminus. So the x-axis is the sequence of the numbers of residues. Okay, so this is 0. Number 1 is the first N-terminal amino acid, and this protein, how long is it? It's about 130, something like that. So this is the sequence down here. And what you do is, let's start here, sort of in the middle here, because that's easier to talk about. So we go to this amino acid, and you look at windows of 10 amino acids on either side of that residue. So if we pick, so this, let's say this is residue, let's say it's residue 20, okay? And so what you would do is residue 20, and you take 10 amino acids preceding it, and 10 amino acids in front of it, and then you average all of those hydropathy indexes together. And you get a mean hydropathy index for that window centered around residue number 20. And you ask the question, what's the average hydropathy of that whole window? Okay? And if it points down below the 0 axis, it's polar, it's hydrophilic. If it points up, it's beginning to get hydrophobic, right? And so we're going to take that window, and so that's how we got that number right there. Okay? And then you move over the window 1 residue. So 10 before, 10 after, take the sum, or take the mean, and look what's starting to happen. It's starting to get even more hydrophobic, right? But what happens when if suddenly the window starts to move into this region of the protein? The reason this starts to climb up is the window is getting in more and more amino acids that are hydrophobic. And when we center that window right in the middle, this is the middle of that alpha helix, it's going to have 10 on this side and 10 on the other side in front of it that are all hydrophobic. And so that's why this peaks right there. So this little hydrophobic peak should be the same number of amino acids is required to get across the membrane. So we look for peaks that are, number 1, positively charged, hydropathy averages, and number 2, they have the correct length. You have to have both. But out here, this is glycophorin. Clearly, the N-terminus is cytoplasmic. We can tell that. And here's the alpha helix, okay? And that makes it across the membrane, and so then it goes into another hydrophilic region, so it's come out the other side. So we can actually tell which side of the membrane or which side of the protein is going to be facing which direction. Okay.
How can you tell that with the N-terminus and the T-terminus, like with the inside and outside?
Probably, let's think about that. Let's take a look at. Actually, I think you're going to need. Let's take a look at the structure of glycophil and see if we can do that. You may have to have. Actually, the way that you can figure it out is where those sugar residues are attached. Let's see if we can. Here it is. I think I spoke too early because I just know that that's the N-terminus. So you wouldn't know, would you? Right? I mean, if you flip this around, it would have the same hydropathy index, right? If something's outside, you wouldn't know which piece is outside, which piece is inside without additional information. So that's a good point. I knew that was in there, so that you can't really tell that from the hydropathy index. And so that's a good reason why you need those other things to help you out. You look for disulfide bridges, and you look for attachment sites. So there's actually a consensus sequence where you would put the sugar, so you can look for that. And that would tell you which side is outside. Yeah, thanks for bringing that up. Okay. Any questions about how that works? Sometimes students get confused with that. So could you do that with a multipass membrane protein? Absolutely. We've talked about glycophorin, right? That's what glycophorin, or I'm sorry, bacterial rhodopsin should have 7 peaks. Now, it's not as clean as you'd like it to be. In other words, you have stuff like this, but there are programs that help get rid of the noisy parts. And essentially, all of these meet both the criteria of being very positive, having a peak that's sort of in the middle of a 20 to 25 amino acid segment. Now, let's get into, more briefly, into membrane dynamics. And that's about what determines fluidity of the membrane. So we have some real common things to look at. So proteins diffuse laterally in the plane of the membrane. Whether or not they're lipid anchored or they're transmembrane, they're going to diffuse in 3 dimensions, or 2 dimensions. They're not going to flip-flop. That's one thing they don't do. They don't flip-flop. And but they can move very fast, and the lipids are moving as well as the protein. So the protein is moving through this sort of goop of oil, and the fatty acid is moving in 2 dimensions, but it's also spinning rotationally, very, very fast. Okay. But one thing that these fatty acids or these phospholipids don't do is they rarely flip-flop from one leaflet to the other. That's energetically unfavorable. Does it ever happen? Absolutely. But it's rare. And actually, we have proteins that will do that. We have proteins that will flip-flop lipids from one side to the other. So it requires a transport type of protein. Okay, now, so what are the rules? You should have experienced these before. But if you want a change in fluidity, what's the most basic thing you can do that affects membrane fluidity?
Saturation.
I mean, even the most basic.
Chain link. Chain link. Chain.
Even more basic than that. Because that would require building changing the actual structures of the lipids. What could you do externally that would change the fluidity?
Temperature.
Temperature, thank you. Okay. So, it doesn't matter what system you're talking about. If you increase the temperature, you increase the fluidity of the membrane, all right? Why do we not worry about that? We're homeothermic, right? Endothermic. We have the same temperature unless we're sick, right? So, mammals, endotherms do not worry about that. Birds, okay. But let's just look at that property that's changing with heat. And this is called, is that focus? Yes. Scanning calorimetry. Okay. And so you're looking at heat absorption. And that's like kcals per mole is on the y-axis. And this is temperature, degrees C. We can just put 1, 10, 20, 30, 40, okay. And so each biological membrane will have some melting point, and this is what it looks like. And so before you reach the melting temperature, the membrane is solid. It's in a gel state. But right around the point where the melting occurs, it forms a peak in heat absorption. And that peak is called the transition temperature, okay, the TM, the melting temperature. Right? So it could be 28 degrees. See? Right? And so each type of membrane has a unique melting temperature. And as somebody mentioned, what if you put more not saturated where there's no double bonds, you put more unsaturated double bond phospholipids in there, which way is that going to shift the melting temperature?
Right.
So we're going to put a lot of things with double bonds. And what does that do to those fatty acid legs?
Kinks.
It kinks them, right? And so you can think of sort of the disco hall when everybody's sticking their leg out and spinning around, they're bumping into each other, right? And so that increases the fluidity. And so that's going to lower melting point. So increases fluidity lowers the melting point. if you make those lipids stand there like this, and they got a bunch of people that don't want to talk and don't want to dance, and they stand there like this, packing is tighter, and so that's going to raise the melting. And you're going to have to get it really hot before they'll start moving around. So temperature directly affects fluidity. Degree of saturation, and saturation is about how many double bonds are there in there. If it's completely saturated, that means no double bonds. If it's monounsaturated, it means one. If it's if it's two. So polyunsaturated means there's several double bonds, and so their legs are sticking all over the place. Right? So more unsaturation is more fluidity. More double bonds, more fluidity. So that's just a general principle to remember. And so this guy, this is the kink in there, double bond there. And those double bonds are always cis, right? What's bad about a trans fatty acid? It's not natural. Trans is not natural, it's cis. So that's why that leg sticks out so bad. And what about length? Is shorter or longer more fluid?
Shorter.
Or shorter. Just think about this. Short thing can move through the sieve a lot faster than the long thing. So short tail length is affects that. So let's do this thing. I'm not sure what happened.
I can't.
Okay, we have time. So, so try to answer this by yourself first, and then I'll let you talk to your group. Some of you are really fast readers. Okay, why don't we finish up? 15 seconds. Oops, sorry.
In order to respond, decrease in fluidity and temperature.
Okay. Most of you think it's number 2, but there's a significant number that thinks it's 4, so let's talk about that. And really, you don't have, unless you know about cholesterol, you wouldn't know whether 5 is correct or not. So let's talk about that. Good. So let's talk, first of all, before your group talks, let me mention about cholesterol, which I've already should have talked about. What does cholesterol do to fluidity?
spaces out the lipids, and so it
Decreases.
increases the fluidity.
Decreases.
Does it anchor the layers decrease, or it's a part of the lipid rafts?
If it's in a lipid raft, it is true that fluidity of the lipid raft is less than the external. Let's get rid of lipid rafts for a moment and just think about what's the most fundamental contribution. It's not entirely obvious. Did you? Right, so what she's saying is that it depends. It depends on what the temperature is, okay? And it is, cholesterol is a fluidity buffer, okay? So if the temperature goes too high, that's dangerous, right? And cholesterol will decrease the fluidity in high temperatures, and it will increase the fluidity if the temperature goes too low. So it's a fluidity buffer. Why is that the case? Well, let's talk about the decrease in fluidity. What happens is, at high temperature, the dominant effect of cholesterol is that the head group is not a ethanol, a glycerol backbone with a phosphate in the big head group. It's a single hydroxyl. So it's got a little tiny head that fits in between the other phospholipids. So it allows tighter packing even at higher temperature. That decreases the fluidity of the membrane. Also, that steroid, let's see if I have a picture for cholesterol. That will help. We'll come back to this. I guess I can. Okay. So cholesterol has a nucleus that's very hydrophobic, but it's got 4 different rings, and that's very stiff. And so what it does, the effect of that is that it prevents those fatty acids from vibrating at the rate at which they would otherwise at a given temperature. So as the temperature goes high, the fatty acid legs are going to want to try to rotate faster. But that flat, stiff steroid nucleus prevents that. Okay, so it decreases the fluidity because it has a small head group, and all those fatty acids can pack together. And also that stiff 4-ring, it's actually a cyclopentane phenanthrene ring, stiffens the membrane and prevents the fatty acids from vibrating as fast. Okay? So that's at high temperature. What happens when the temperature goes low, like in this case with the insect cell culture? So we've lowered the temperature. Would cholesterol help out there? Absolutely. It increases the fluidity at low temperatures. And that's the reason for that is attached to that steroid nucleus is a long aliphatic chain. Okay? And that aliphatic chain, what it does is it causes tighter packing. It prevents those highly disordered lipid tails from vibrating so much. Or it increases the aliphatic chain, pushes them apart towards the center of the membrane. And that's why I need a structure to show you. But that aliphatic chain increases the fluidity by disrupting the fatty acid tails down near the center of the lipid bilayers. So it has 2 effects depending on the temperature. So it's going to help out at high temperatures, decrease fluidity. Low temperatures, you want an increase in fluidity. Cholesterol helps out with both of those. So answer E would not have been the correct one because cholesterol helps out in both directions. So let's go back to this. Okay, now talk to your group and figure out what. So the battle seems to be between this thing and this thing. And then have your, let me open the polling again.
Longer chain, longer tails decrease fluidity. Unsaturated fatty acids.
Because if you would shorten the fatty acid tail, that would, yeah, but on me it's saying you're making the tail longer, so it's making it less fluid.
So
The problem is that less fluid, so you should really do the opposite.
I don't know. I can't think right now.
Make it shorter.
Right, but what is happening to someone?
We're still in the limit.
It says when it's off.
Yes?
Yeah. So bees only when it's on.
If you what? We can do that really quick, yeah? She's not gonna do that. We pick B. Get out. Or not. Get out or not. So we can both make it. That way we don't have to wait in line.
Yeah.
Yeah.
Like the chili cheesesteak place.
Right.
You should. So it's getting more and more packed. And it's like, yeah. Like I think one day Chick-fil-A took me like 15 minutes. It's like an express line. So it's like, yeah, buffet. Okay. Yeah. Because you're like, oh sure, whatever.
Yeah.
Yeah, we definitely don't have 20.
Okay. All right, now we have a much more inclusive. Good, so. Yeah, so one of the things you don't want to forget is the basic effect of temperature. It always directly affects fluidity. And so if you lower the temperature of the cell culture, it immediately is going to lower the fluidity. But then the cells are going to try to respond to that fluidity challenge by doing certain things. And certainly, one of them that you would not want to do is to replace the short fluid-making fatty acids with long fatty acids that make it less fluid. Because you're already cold, you don't want to become less fluid. All right. I think we'll stop there, and we have the test on Tuesday.
I didn't think about it. I was just read the
Listening to the lectures while studying is really helpful. I listened to lecture 1 and most of 2. It's really nice. Like they have talks and I'm going through your notes and memorizing it. Why is this class always in a hurry? It's like I'm never in a hurry to get to class on time. It's like biology for non-majors too. Yeah. I want to learn stuff I don't care about. Did you notice me sleeping in the class? Did you notice me?
Yeah. Right there.
I was like, what
I did. I was like, I don't fall asleep. I'd close my eyes and then I'd open them. I thought he was gonna put. I was like watching him throw something at me. After that break. We're studying tomorrow, right? Tomorrow. Yeah.
Okay, good. I'm going to make sure that I go. I still haven't listened to the train.
Oh,
So,
uh-oh.
yeah, no, I took that GRE diagnostic test. Literally, right after my genetics test, I went home and slept for like 4 hours. And then got up, took that test. The next morning, I got a phone call from the Kaplan guy. And he was like, yeah, we've decided to postpone the class a week. And the days are going to be Wednesday and Saturdays now, not Monday in the morning to take a diagnostic test so that you could call me the next morning. I didn't really take it yet. So I wasted all that. I mean, I could have done more. So, but I started lecturing and I haven't finished it, but I haven't done that in my
See you tomorrow. So on the jacket. I'm so bad about, you know, I'm so bad about like, oh, what clothes did I wear for the photo? When I'm doing better, I didn't wear shorts. I got ready with shorts, and then I looked outside, I was like, well, I'm getting better. I realized I do that. You're sorry, library, right? I realized I do that.
Well, you don't have to go back to because I just have to draw straight lines. That's all I have to do.
Oh, okay. Oops, I've been recording ourselves talking.